Hey Sheila, since I know you personally very well, but I want you to share who are you and tell a bit more of yourself to our audience.
Well, I have been a biotech entrepreneur along with Jude Samolsky, who everybody knows. Jude was the first to clone AAV for therapeutic purposes for about 25 years. My background is I'm a lawyer and I have a finance background, and how I got involved in gene therapy is a very interesting story. I was working in my law firm, and one day a professor at UNC called me up. He was teaching a course called Launching the Company, and a speaker was not able to attend. And I guess that day's session was supposed to be about spinning out companies from the university. And he said, would I mind coming down, it's just down the road, and filling in for the speaker? And so I did. I went there and I talked about what you need to do in order to start a new company and how to negotiate with the university and the license agreement. And Jude Samolsky was in the audience.
Okay.
And Jude came up to me afterwards and he said, I have this incredible idea. It's called gene therapy. I didn't know anything about gene therapy.
Of course, at that moment, yeah.
And this is like 2001.
Yeah.
And he said, a lot of people are skeptical. I believe it's going to work. It's going to take some work, but eventually it's going to work, and it is going to save children's lives, because a lot of these kids do not have any treatment. They have genetic diseases, and they have a very bad outcome. And he goes, I just want to be able to see if I can do something. And there's only so far I can go in an academic setting. I can't take the drug into human clinical trials, so I need to form a company. And he said, will you help me? And he also said, I probably won't be able to pay you, which he didn't for a very long time. But he was so genuine. Anybody knows Jude. He's very easygoing, and he gets you very excited about what he's working on, and he makes it so easy. I didn't even know what gene therapy was, and he said, well, think of it this way. I'm creating biological FedEx trucks, and we put little zip codes on the packages,
exactly.
And we tell the biological FedEx truck, called an AV vector, where to drop off the package. And once it's there, then the therapeutic gets created using the body as its own bioreactor. I'm like, okay, that's common sense. It makes a lot of sense. I think if he can make it that simple to understand, then I'll help him with the stuff I know, the finance and the legal. And we've had almost a 30-year partnership where Jude does the science and I do the business and the legal and the deal structuring.
Oh, great.
So that's my background.
Oh, love your story. Yeah, this part I didn't know. But yeah, it's it's hard actually, very challenging to find a partner and it can go that long, 30 years, like hands hold hands. And then I believe a lot of happened during the journey, but you guys make it through.
And we made it through a lot of tough times, as you know. The field has been up and down, a lot of cycles. And I think even now we're in a tough position now. Right. But it wasn't as bad as it was in 2000 because we have had some wins. We have approved products. So, it will come back. There's a lot of turbulence politically, economically, research-wise, lots of things going on. But I think we just have to stay focused on what we're doing and solve the problems associated with AAV and demonstrate that we have solutions.
Right. Yeah, that's also my second question. So now it's kind of a little hard for especially AAV-based gene therapies. And then the investors probably, try to ignore, when you talk to them and they are focusing other pipelines or something. But then what's your view for AAV-based gene therapy specifically? What could be the breakthrough to bring this stuff, AAV gene therapy, back to the market again?
So I think that there are several things that can be done to really make sure that gene therapy is successful in the future. The first one, as I showed today during my presentation, we have to have better capsids that have improved potency, but also improve safety. And we're using the human decedents essentially to demonstrate those capsids, to find those capsids, right, and to target particular tissues. So that's number one. The second thing is it would be great if we could actually get a regulator system to work where we can turn on and off the transgene expression. That's beneficial for two reasons. One, you have a kill switch, so the safety then, you don't have to worry about if a drug isn't working well. The big concern now with AAV gene therapy is, well, you can't turn it off. Right. Well, no, if we use a regulation system, a regulator, then we could turn off that transgene expression. But I think also there's a practical side, and that is that we can fit into the business model that big pharmaceutical companies want, and that is that recurring revenue stream where a patient pays, smaller dollar amounts, but pays repeatedly over time. Whereas today what happens is there's a big price tag and the payers aren't certain about this, they push back. And that's why I think you see some of the other transient molecular medicines pushing forward, because it fits better in the paradigm, the commercialization paradigm of big pharma. And then I think the third thing, of course, if we could, and I think there are technologies out there with the antibodies, we demonstrate some initial proof of concept, but if we could further develop those antibodies to expand the inclusion criteria. Right now, as you know, for most of the endogenous AAV, there's an exclusion criteria that could be up to 40%. 40% of the patients aren't eligible for a specific, capsid type. If we could, use the antibodies to include those patients, expand that market size, the additional 40%, that would be very beneficial. But also explore other antibodies. And again, there's mechanisms out there. We know this. And if we can adapt those developed antibodies for use in gene therapy, where we could do repeat dosing. And again, there's so much work being done in autoimmunity and whatnot that if we take those learnings and apply it to gene therapy and collaborate with antibody developers, I think we will figure this out technically. That will be a huge win because, again, mostly because it will fit into the paradigm of commercialization that big pharma likes, that recurring revenue stream.
Great points. Yeah, I think those are the biggest challenges, roadblocks existing there to block our way. But if we kick them off and with the new technologies and platforms and fundamental changes, AAV can be back again into their stage. Great. So being as a successful entrepreneur, I want you to share some advice to those beginners, to those scientists or researchers who want to be follow your path. Any suggestions?
So I would say in today's world, the paradigm is you must use Chinese IIT. You just I developed one drug for Pompe here in the U.S. It didn't work. I had to redesign it. You developed it overseas with Dr. Xiao, Believe Bio. It will cost significantly less, 40% of the cost, much faster, and the skill set, right, in China is amazing. Being able to translate it into Chinese IIT, you get that human POC, proof of concept, so much more quickly. So, it's very upsetting to see some of the government policies about collaborating with China because honestly, I think it's the only way forward. It's the best thing for patients. Collaborating with China lets us decrease the cost of drug development, get drugs to patients who need them much faster. And honestly, a patient doesn't care if a life-saving drug is developed here in the U.S. or in China. They just don't care. And I think the Brits have figured that out because they're now accepting data generated in China. And I hope that the U.S. understands that too. We shouldn't be enemies. We should collaborate.
Together, right? Like for the whole world, the whole population of the patients, we are in the same community actually.
Exactly.
Yeah, great.
Exactly.
Yeah, great to share your insightful thoughts, and I totally fully agree about this. So I think the last question is like, what do you want to say to our SAPA-GP audience, and also what kind of other improvements that we can do for our event for this CGT meeting in the future?
I love the event. I love coming up to Philadelphia every year. I love seeing old friends I haven't seen for a while. I love the idea that we have some planned trips to China. I think it's really important to build those bridges back to China and to really make it more accessible for more American drug developers to find collaborators in China and to really, put together our resources and our knowledge and try to, advance things for patients as quickly as possible. So I would just say keep doing that. Keep partnering Americans and Chinese collaborators together, because I think China has right now a lot of exciting things going on. And I know a lot of my American colleagues would love to participate, right?
Yeah. Welcome back. Actually, right now I'm working in a Chinese company now, and even though we have site in the Philly, I myself is based in Philly, but my company is in Hangzhou actually. So welcome to China.
I like Hangzhou. I like it very much.
Really? Great, Xihu, right?
Nitrogen is based there.
The Westlake, yeah.
Westlake is beautiful. I
right?
stayed there.
Yeah.
It's absolutely beautiful.
Great.
I was there in the fall, and it was just lovely.
Yes. And thank you for all the good words, and appreciate your contribution, and also all the efforts for this field, for this community. Thank you so much, Sheila.
Thank you.
Thank you.